Hi class, welcome to our lecture, Methods in Biological Psychology. For this lecture, our learning objectives are to describe and distinguish between correlational and experimental studies, discuss important issues that modern behavioral neuroscientists must contend with, such as the use of animals in research and the replication crisis, and explain the different levels of analysis that may be focused on by behavioral neuroscientists. Biological psychologists use three general approaches to study brain and behavior: the somatic intervention, behavioral interventions, and correlational studies. I will describe each of these in turn. With somatic interventions, we alter the structure or function of the brain or body to see how this alteration changes behavior. For example, we could administer a hormone like oxytocin and see if participants who receive the hormone show more empathy towards others compared to those who do not receive the hormone. In this example, the intervention, the oxytocin administration, is the independent variable, that variable which the researcher manipulates. The behavioral change, like empathy, is the dependent variable, the variable which we must measure to see if our manipulation has an effect. Other examples would include stimulating a particular brain region to see if there is a change in movement towards a goal object, or cutting connections between parts of the nervous system to see if it influences the recognition of a stimulus. An alternative approach is to use a behavioral intervention. With the behavioral intervention, researchers manipulate the behavior of an organism and look for resulting changes in body structure or function. For example, researchers could attempt to induce empathy in participants, perhaps showing them videos of others suffering, and then measure the level of circulating oxytocin in the participants. In this example, researchers manipulate the behavior, empathy, and measure any resulting changes in hormone levels. In this case, behavior is the independent variable, and the bodily changes are the dependent variable. Final approach used to examine brain behavior relationships is a correlational study. In a correlational study, the researchers make no effort to manipulate any variable and is only interested in seeing if the relationship naturally exists between variables. For example, you could examine if a relationship exists between circulating oxytocin levels and early childhood maltreatment. Here, you wouldn't be able to determine cause-effect relationships, a particular limitation of correlational designs. However, demonstrating a correlation can be an important first step in exploring brain behavior relationships. Biological psychologists also use several levels of analysis in their studies. Because of the complexity of brain and behavior, we often attempt to analyze behavior on a simpler or more basic level of organization than that being explained. This approach is known as reductionism. At the broadest level, we're interested in how individuals behave at a social level. The newer field of social neuroscience attempts to understand the relationship between brain function and social behavior, the previous purview of social psychology. We next have the level of individual or organism and the nervous system as a whole. If we look closer at the brain, we can examine specific neural systems and particular brain regions involved in specific processes like vision. We can continue down to the circuit level, the cellular level, the synaptic level, and the molecular level. Each of these levels of analysis, of course, are interrelated. For example, in some of my own studies, when the developing brain is exposed to alcohol, a number of molecular alterations occur, including changes in molecules, that is the molecular level, in areas of the brain important for learning and memory, that is the neurosystems level. And these alterations are associated with learning and memory impairment, that is the level of the organism. We can note here that some of our more invasive techniques to study the brain, and especially at a deeper level of analysis, makes working with humans problematic for ethical reasons. And researchers and the neuroscientists have often used animals to model human behavior and various disorders so that they can better understand the relationship between brain and behavior. One very famous example is from the Nobel-winning neuroscientist Eric Kandel, who was interested in studying memory processes in humans. But to understand that, he used a simplified animal model, the Plesia californica, the sea slug. So in this particular slug, he was able to identify individual neurons. They have a very simplified nervous system that were involved in a particular form of learning and memory. And in that simplified form, because he could physically identify which neurons were interacting with one another, he was then able to examine the molecules and the molecules involved, the changes at the level of the synapse, all the way up to the changes in behavior. We'll be talking about the use of animals and the relationship between that and humans throughout this course. I do want to identify a couple of modern techniques that you will come across in our studies. I'll refer to these when they become apparent in our discussions of various topics. So, one such technique that has revolutionized the study of the brain are just modern advances in imaging techniques. Starting in the 1990s, late 1980s, was the development of functional magnetic resonance imaging. This imaging technique takes a very detailed picture of the brain, and by examining changes in blood flow can identify those regions of the brain that are more energy expensive. Therefore, those areas of the brain are active during particular behaviors. So we can use the fMRI in order to understand which areas of the brain are active during particular behaviors. Newer techniques called diffusion tensor imaging, or DTI, allow for a detailed examination of the connections between cells. What we're looking at in this image on the right is a DTI image. This is the corpus callosum, that brain region that spans the two hemispheres, allowing connections between the two. So imaging techniques. Genetics. It seems like a very long time ago, but back in 2001, both Science and Nature published a complete mapping of the human genome. In fact, I have a copy of the Nature edition. And the advances in genetics have been well advanced over the past 19 years, including techniques like CRISPR, where we can go in and use a molecular pair of scissors to cut out and edit genes. These techniques are allowing us to do amazing things to better understand how genes interact with environmental processes and aid in our understanding of the biological basis of behavior. A technique that I have been quite active in is the use of optogenetics. That is the combination of using lasers and genetic techniques in order to understand brain function. So in this technique, we use a virus in order to convince neurons to express cells that turn on and off in the presence of light. They act as light switches. And using some other techniques, what we have done is have certain cells express this molecular light switch, but only during periods of learning. Therefore, we can use a laser to reactivate a subset of cells that happen to be engaged in a learning process. And thus we can artificially reactivate memories. We can also inactivate memories or erase memories. So if you're thinking of Eternal Sunshine of the Spotless Mind or Total Recall, some of the movies where they manipulate memories, just know that in current neuroscience labs, we are actively doing this, at least in animal models. Here's a rat that is expressing a fear memory, not because they are afraid, but the laser light is activating cells that had been active previously to a fearful stimulus. Each week, we're going to have a series of what I call retrieval practice questions. They're typically embedded throughout these lectures. I'm posting them here, but here are 4 questions that I would like you to answer, and then submit your answers on Canvas. First, what is another name for biological psychology? We went over a couple of different synonyms. This class is an introduction to biological psychology. Name at least one other name for that. 2, identify the researcher that identified that nerves conduct information via electricity. Was that Cajal, Broca, Galvani, or Lowy? 1.3, what area of the brain did Descartes postulate to be the place where mind and body interact? Was that the hippocampus, the pineal gland, the ventricles, the brainstem? And then 4th, what is something that you would very much like to know about the brain? And here, I just want you to think about something that you're really interested in or you really want to know. Put that as your answer. I'll read through these and try to incorporate some of what you want to know into this class to make sure that you're getting the most out of this class.